Well, welcome to the Multiverse Theater at Mozak. How about a big hand for this beautiful museum? My name is Patrick Desmond, and I'd like to thank you for joining us today for Sacramento Darwin Day's 26th anniversary. We firmly, go ahead and applaud. We firmly believe, until proven otherwise, that this is the second longest running Darwin Day celebration in the world. It's pretty cool. And we stuck it out through COVID and all that. It is my honor to serve as the chair of the Sacramento Darwin Day Organizing Committee. Members of the committee are dedicated and passionate individuals dedicated to promoting and supporting science in the greater Sacramento area. It is our goal each year to find an exciting and compelling speaker who can further our understanding and enthusiasm for science. This year is no exception. I believe you'll find Dr. Rachael Bay's presentation to be timely and informative, since she is at the forefront of her field. In addition to Dr. Bay's presentation, one of our esteemed founders, Minga Futrell, will provide her annual presentation on the importance of this event and why science is vital to our communities. Before I introduce Minga today, I'd like to take a moment to thank all of you in attendance, as well as those who have supported us through this year's but could not attend. We have a lot of people that donated and bought tickets that couldn't make it, but they wanted to send their regards and their support for everybody, I believe, here opinion that science is important and something to be promoted and enjoyed and appreciated. And we as a community have its back. There is no need to dwell on the strange and challenging times we have all endured since February of 2020. But despite these challenges, Sacramento Darwin Day has come roaring back with new vigor and excitement and a new home here at Mozak. Thank you. Our sincere gratitude goes out to you for your support, patience, and understanding during these challenging times. You had mentioned you wanted to see some old friends here. Did you see some of your old friends? Oh, that's right, you don't hear me. He came up, he wants to see some old friends. We see old friends here. Yes, you see Minga? Minga, where you at, Minga? She'll be up in a second. There, she's right behind you. She's right behind you. She'll be up in a moment.
She's over there?
She's right behind you. She's very small. Darwin Day in Sacramento will continue for the same reasons it was founded 26 years ago by Bronda Silva. Bronda, where are you? Next to Minga. Hi, Bronda. So I did the program. Bronda's S, Silva's last in the alphabetical order of our committee members, so it got pushed off. My apologies, Bronda. She's so important to our committee. She's our treasurer and just been a real rock star for us. So I'm sorry her name should be in that program, and it's my fault. But thanks again, Bronda, for everything. I hope you're happy with how everything turns out. The world of science is essential and endlessly fascinating, and it deserves recognition. Our circle of friendship expanded greatly through our collaboration with the fine people at the SMUD Museum of Science and Curiosity. Our community appreciates science and science education because of the talent, expertise, and passion of these fine professionals. I hope you all had the chance to walk around this amazing and beautiful new facility before the presentation, and that you will do so again afterwards. I'd encourage you to join me and become a member of the museum. This gesture will benefit science and will serve as a vote for it. I'd like to thank a few people personally for making this event possible. Museum of Science and Curiosity Board President Michael Gillane. Michael spoke at an earlier Darwin Day celebration, and many of you supported the Capitol campaign to build this beautiful building that year and every year since. I'd also like to thank Michelle Wong, the Executive Director of the SMUD Museum of Science and Curiosity, for believing in us. Congratulations, Michelle. You've done a great job and you've built a beautiful building. The community is better for it and your hard work is appreciated. I'd also like to thank Jennifer Martin, the Director of Experiences here at the museum. She was the first one to take my call, and she helped champion the idea of bringing this event here. I am going to also thank afterwards, but first I'd like to bring up, to say a few words, the Mozak Director of Marketing, Rod Malloy. Rod? There he is. Big hand for Rod, please.
It's been a pleasure of mine getting to know Patrick over the last month or so, and we found out we have a couple of commonalities in terms of the world of filmmaking, and I'm going to watch Patrick's film tonight when I get home. But I want to point out something that is in your program. It's a sheet of paper that's marked Legacy Giving at MOSAC. Patrick and I decided to insert this into the program just to give you a little bit of some tips and my contact information in case you want to make that courageous decision to add this museum and this organization to your estate plans. It's interesting because the organization was founded in 1951, and it has had several different names over the years: Discovery Center, Powerhouse Science Center, and now the Smug Museum of Science and Curiosity. When you think about those 71 years leading up to today, or last year when the museum opened, think about 70 years into the future and what first and foremost, your membership within the museum, but also your generosity in the years to come. It will impact thousands, hundreds of thousands of lives. I read a couple of children's comment cards from their experience this weekend, and it reminded me of my experience as a young boy going to the Hayden Planetarium in New York. Our person who works here and operates the dome, Jeff, he also grew up in New York going to the Hayden Planetarium. Now we have a whole nother generation of young and middle-aged and old people who are enjoying this museum 7 days a week, 363 days out of the year. So it's a phenomenal organization. It's an honor for me to be here tonight for Darwin Day, and I appreciate all of you for being here and for your support of Darwin Day and the museum. Thank you very much.
One more thing.
You have a gift in our gift shop.
Oh, yeah.
Do you have yours on you?
I don't.
So you're going to get a bust of Charles Darwin that is about this big, and there are different colors, so don't fight over the colors, please. There's a lot of white ones, that's appropriate because he was a white gentleman.
Thank you. Thank you. Big hand to Ron, please, and everybody at MOSAC. Yeah, it's a 3D printed bust of Charles Darwin, and it's a little bait to get you into the gift shop. So my master plan is that we found a new home here at MOSAC, and just to be fully transparent, I think it's fantastic. Now, seating's limited. The quality of this event is very high, and I think it's going to sell out in the future once word gets out. We have about 80 people in here. We can do about 117. We add some chairs for presentations. So my vision is for this event to grow in quality and substance, and maybe it'll outgrow this room eventually. You can see there's a lot of room out there. But the more we show up tonight and support, make donations, become members, and appreciate the gift shop, the more they're going to understand that, yeah, this is our community, these are our people. And that's what I told them years ago when I came to them and I said, we should be working together. And now we are, and I'm just super thrilled. And thank you again, Rod. I wanted to make sure that I recognize a couple other people here not able to be here. I wanted to recognize Bonnie Patterson couldn't make it. She's the Director of Finance and Operations, and she was a huge help, and I want to thank her. And then Susie's an event coordinator here. She's doing a fantastic job. And Forrest in the back is running his beautiful planetarium. He must be very proud of it. You've got to come back and watch the shows here. There he is. And check it out. Please do. Each and every one of you is appreciated, and I look forward to another fun event next year. I would like to thank each of you for purchasing a ticket and for your generous donations. All donations to Darwin Day, all your ticket funds, go towards providing this event to you, supporting science and providing speakers who join us each year. All committee efforts are voluntary. Thank you again. It is now time for one of my favorite annual traditions here at Sacramento Darwin Day, the traditional celebration of all things science. Clearly, science and the scientific method are the key to our survival as a species, and they need to be exalted, defended, and discussed as often as possible. Dr. Minga Futrell, a founder and past chair of Sacramento Darwin Day, is here to remind us all of why we celebrate today. Minga, please come up. is.
Except I won't spit in it, I promise you. That's scary.
There you go.
Oh, good evening, everyone. I am delighted to see you here for Sacramento's 26th birthday celebration. It's hard to believe. Our volunteer organizing committee, it's kind of evolved over the years, but it's put on so many. And looking back, I'm afraid I've been part of them for more years than I'd like to remember. But I almost, but not quite all of them. Brenda Silva actually has me beat by just a little bit. She's among the current committee members, even though she was left off the program. And who are we, the people who've been putting this on? We're just simply science enthusiasts. We want to toast science for its benefit to all of humanity and learn some cutting-edge science for ourselves. A little self-interest in here. And we want to commemorate the approaching birth anniversary of someone who was and remains a scientific giant. So, for all you bipedal primates that have shown up here this evening, we'd like to add a bit of hoopla to this learning experience. Folks like us are not alone in wanting to celebrate the life and legacy of Darwin. There are events being held in colleges and universities, as well as community gatherings elsewhere in the nation and even across the world. Much of it, lectures, the sermons, museum exhibitions, film showings, parties. Very few held as early or, as they said, as continuously as our own here in Sacramento. Of necessity, we had to do ours online for a couple of years, but we made it through. We're excited to be back in person to take note of Darwin's immense contribution to humanity. One year, my annual 5 minutes message, I spoke of Alfred Russel Wallace, who many do think really should be co-honored because he did sort of scoop Darwin in terms of his thinking about the mechanisms of change. But it was Darwin, with Wallace kind of hot on his heels, who finally set aside concerns about how his ideas would be received by the public, and finally, 20 years after reaching them, published his The Origin of Species. It's because of that timing that we don't remark on Wallace Day today. The publication was a singular sort of event, revolutionary in educating the citizenry about how nature works, right here at home on planet Earth. It made quite a splash, although not necessarily a popular splash at the time, or even now with some people. Science has definitely taken off since Darwin and Wallace put forth their mechanism of evolutionary change. That daring idea of natural selection has had and does have a permeating effect on our own lives today in so many fields of science and all across the world. Contemporary scientists deem it the most powerful scientific theory ever developed. Tonight is an educational occasion. We all will, without doubt, go home tonight having learned things that we didn't know before, exulting in some, perhaps despairing of others. But this evening will be educational. We will have learned. At this event, we welcome science enthusiasts of every stripe, but particularly educators. So I would like to give a special welcome to the representatives here tonight from Sac State's Department of Biological Sciences and its Anthropology Department. We appreciate the endorsement of these college-level science departments because we who put on Sacramento's event have long had an interest in promoting and defending not just science, but also sound science education at all levels. We seek to maintain authentic science education in the face of creationist movements, to advance scientific understanding in the face of irrationality, misinformation, and conspiracy theories. We care about science. And much of the public today ignoring science, even aggressively countering it, is troubling to us. Politicians undermining support of scientific inquiry, failing their constituents. That's worrying. We have particular concern about public school programs. Learning at one point that less than a third of school biology teachers in the U.S. explain evolution by natural selection in a straightforward way, presenting evidence for evolutionary change as recommended by the National Research Council. That's truly disheartening. And to learn that 15% still are explicitly advocating creationism, that's truly disturbing. Do we ever need better scientific understanding by the public and recognition of the importance of active science to public welfare? Seriously, folks, with climate change quickly gaining on us, and some lawmakers still, yes, still denying the science of it, a citizen with a good grasp of science and appreciation is the strongest defense against the unsettling future we face on this planet. But we also call this event not just education, we call it a gala. And the main program may be to educate, but it's a social event too. It's going to turn into one. Watch out. Besides turning your brain onto science with the formal presentation we're getting ready to hear, we really want to show appreciation for Darwin's big idea and to reaffirm it as momentous by singing Happy Birthday, the song in praise. It's a tradition with our gala, and don't leave without that special gift. So here's my message. To all you champions of evolution and science this evening, let's celebrate. This isn't just a lecture. This evening is a celebration. Let's celebrate science. Science belongs to everybody. Its methodology has been a true gift to humanity. Evolutionary theory has become immensely important to increasing human comprehension of the natural world. And let's celebrate that fellow Darwin himself. He has become a symbol of scientific advancement that can and should be promoting a common bond among all of Earth's people, not divisions over ideology, especially the ones that continue to impede educational progress. Plenty to be celebrating about that person too. Everyone talks about Darwin, and rightfully so. In life, he was, by most accounts, in addition to being a fabulous naturalist, he was a kind man, all around a pretty nice guy. So while you're here, you be nice, you be sociable, chat with others, be sure to eat up some of that birthday cake. It may be his birthday, but he's the one that gave us the gift. It's his gift to a better understanding. But the current social and political context, we've got to depart this event to work harder than ever to support and preserve sound science as a hope for humanity. And we need allies in our desire to succeed in that preservation. They're out there, so next year bring your friends and your family, perhaps a coworker or a neighbor. But for now, I want you to enjoy this evening, and let's direct our attention, eyes, and ears to what our speaker has to say about her activities and how they play into the story of life on Earth, as it has been and what lies ahead. So it's time to learn.
You heard the lady. Have fun, dang it. Minga catches anybody not having fun, she's going to make you have fun. Thank you, Minga. Wonderful. We need to publish those. Your presentations every year are fantastic. And I'd like to publish them. I guess I'll do this real quick because I think we're going to have to lower it again. But I want to do a good job. It is now my distinct pleasure to introduce today's featured presenter. This year's featured presenter is Dr. Rachael Bay, Principal Investigator at the Bay Lab. The Bay Lab in the Department of Evolution and Ecology at UC Davis studies the ways in which human-induced environmental change alters evolutionary trajectories using a combination of ecological and physiological experiments and genomic data. Dr. Bay holds a BS in Marine Science from the University of Miami, a master's in science in biology from Dalhousie University. I hope I said it right. I meant to Google that. A PhD in biology from Stanford University, which I can pronounce because it's world famous. Dr. Bay says that she is interested in how human influences on the environment alter the evolutionary trajectories of organisms. She uses a combination of ecological experiments and genomic analysis to identify genetic variation underlying adaptation in natural populations and to understand how adaptive variation is distributed across environments. She is particularly interested in developing predictive genomic frameworks that can be used to estimate the potential for adaptation to future environmental change. Dr. Bay says she has a particular fondness for marine systems, although she has worked in a broad range of taxa, a taxonomic group of any rank, such as species, family, or class. I needed that. Dr. Bay grew up in a multiracial family in a tiny town in Montana. From an early age, she developed a love for animals and nature and was privileged to have a parent that prioritized education. She believes that all people should be able to pursue their scientific dreams and is committed to creating a supportive and inclusive lab atmosphere. In her spare time, she enjoys baking, hiking, and spending time with her husband and her young child. If I may, I'd just like to note how fortunate we are to have such amazing institutes of higher learning throughout Northern California that support such amazing, talented, and hardworking people like Dr. Bay. Some sitting in this room tonight. You make life better for all of us, and I just want everyone and anyone here that works or has dedicated their life to science to know how much your work is appreciated. It is my distinct pleasure to welcome to the 26th Annual Sacramento Darwin Day celebration, Dr. Rachael Bay.
Yeah. Okay. Hi, everyone.
Hey.
Thanks for the participation. It's way better than my intro bio class already. I first want to start by just thanking the steering committee. It's so special to be here with you today, celebrating science, and in a venue that, as a parent of a 3-year-old, I've become very fond of, especially with all the rainy days recently. Today I want to talk to you about how this theory of natural selection, which is over 150 years old now, is become especially relevant in today's climate. So thinking about how natural selection is relevant to climate change. All right, so for hundreds of years, since sort of, I guess, the dawn of humanity, humans have been interested in the variation that they see on Earth and had questions about it. Why are there so many species? Why do we see so much variation within species? Why do some species seem especially well adapted to their environment while others seem pretty poorly adapted? And people have been trying to answer these questions for long before Darwin came around. Some of our earliest natural history writings that are preserved came from Aristotle in 300 BC. And Aristotle sort of adapted this religious view at the time that placed everything in the world in this very hierarchical system where these categories of things, including animals and plants and minerals and people, were in this sort of firm ladder, and that you could not change in position across that ladder, and variation was not on purpose. But not everyone in the world thought like Aristotle at the same time. So Aristotle would have looked at this variation and thought that one of these forms was the ideal form meant by God, and the rest were some sort of mistake. But at the same time, we have Chinese scholars that thought that species changed into other species in this sort of circle, ever-evolving circle of life. They didn't think of evolving as Darwin thought of evolving. We had no mechanism for creating new species, but you have a really different view of the world when you think that some species change into other species. And in fact, when you look at some of the Islamic scholars in about the 8th century, you start to see hints of descent with modification. So some species change into other species, and their traits change to adapt to their new environment. But still, we don't have a mechanism for evolution until Darwin and Wallace come along, and they present their theory of natural selection. So, I can't. You can just click forward a couple times, and then we'll walk through the slide together. Okay, so just to get us all on the same page, how many of you took sort of an intro bio or high school bio class that taught you about natural selection? Okay, great. We're all cool here then. All right, so just to remind you, this is how natural selection works. We start with a population here on our left. In this case, we have orange and black mice. And the key here is that there's variation in that population. That's very important. We can't have natural selection. We can't have evolution without variation. And then we have some sort of selective pressure, in this case, a bird of prey that comes and picks off some of the mice. And the key here is that there's differential success of those mice depending on a trait. So the bird sees some orange mice that stick out more against their background, preferentially eats those. And then the other key here is that black mice give rise to other black mice. You have inheritance of this particular trait. So the important things here again are diversity, so that variation in that trait, differential success depending on the trait, and inheritance of the trait. So that all leads to natural selection. Okay, yeah, great. Okay. So what I want you to take home today is diversity is the key to adaptation. And thinking about, especially when we think about all the global changes that are going on today, thinking about where the diversity is and how important that is for future adaptation. Okay. So, of course, we know that Darwin came up with this mechanism for natural selection, but Darwin was also thinking about this constantly changing environment. People weren't really thinking about climate change at the time, but he did recognize that environments change on a number of different scales. And we can come back to that thinking when we're thinking about really contemporary issues of rapid climate change. So of course, since it's a celebration of Darwin's birthday, I wanted to really sort of present what he contributed to both natural selection and the discourse on climate change. Okay, and in these ever-changing environments, we're seeing this happen more and more rapidly across the globe in a number of different ways. So we have record-shattering droughts. Of course, in California, we're all familiar with this middle picture. And in the tropics especially, we have increasing frequency of storms. So these are all things that sort of happens, but they're happening more severely than before. And organisms living in these environments are subject to new conditions that they've never seen before. And so what happens in terms of evolution when we have these new environments? This is a map showing sort of annual temperature increase. So how hot is the globe compared to its average? And as you can see, almost every place on Earth is hotter than average, hotter than the long-term average. So when we look at any place on the globe, we see that almost every year now, we see that it's hotter than average. So as an evolutionary biologist, when I think of this, I think of the environment that particular species evolved in. So what environment are they adapted to? And what environment are they living in now? And how is that going to change what we see in the future? Okay. And so let's think of evolutionary trajectories. Most species that we see have evolved over millions of years to a particular environment in a particular place, and they're more or less adapted to that environment. But when we look at things like we see on this graph here, where we have more rapid increases in temperature, more rapid changes in precipitation, all these very rapid changes in the environment, what we're saying is that these organisms are now living outside of the environment in which they evolved. So they're seeing environments which they've never encountered in their evolutionary histories, and they're subject to these new selective pressures. Here's an example of this. How many of you read the news stories about the heat waves with all the dead mussels? I think a couple years ago. Anyone see those stories? Yeah, so if you went out to the coast during those times and looked at the tide pools, you saw thousands and thousands of dead mussels. For those of you who don't know mussels all that well, the ones that are open are dead. The ones that are closed are still alive. So if you wandered around, you saw up to 70% of the specimens died during this heat wave event. So at a sort of first glance, you might think this is really bad, and it is. Up to 70% of these animals died during a single event in a single week. However, again, when you think of this from an evolutionary perspective, this is a really optimal opportunity for selection. Yes, up to 70% of these mussels died, but also 30% of these mussels lived. So in my lab, we're really interested in what is the difference between those mussels that lived and died, and what does that mean for the future? So one of the questions that we have to ask when we're thinking about evolution and climate change is, how long does evolution take? I think most of us think of evolution as a process that happens over millions and millions of years on like geological timescales, and it is. Most of the species that we see today on Earth were created over millions and millions of years. However, that doesn't mean that evolution can't be very fast. You can click through several of these. Yeah. So if you've read On the Origin of Species from Darwin, an inordinate amount of time is spent talking about pigeons. And the reason for that is because pigeons are a really great example of artificial selection. So at the time of Darwin, they hadn't really connected the process of breeding animals like pigeons or livestock that was sort of general practice at the time with the processes generating biodiversity in nature. So pigeon fanciers or pigeon breeders would selectively breed pigeons with particular traits in order to make this sort of crazy diversity that you see here. And you see some really exaggerated traits in these pigeons. And all that means is that humans are deciding who gets to survive and reproduce rather than nature, but the process is the same. And we can see the same in some of our crops today. So this is an example of a wild mustard. Anyone know what other sort of common household thing this turns into? Broccoli? Anything else? Cauliflower? Okay, yeah, let's see. So it turns out when you select on particular traits from this one plant, so these are all the same species, you get really different outcomes. So when you're eating kale or cauliflower or broccoli or cabbage, those are all the same species, and they've been selected by people, the process exactly the same as natural selection, to have all these different forms, different tastes, different textures. So the point of all of this is that evolution, if the selection pressure is strong, and the selection pressure that we exert on these crops is really strong, evolution can happen very quickly. And so the main point here is that humans direct evolution. We do this with crops and livestock, and we've been doing this for a really long time. But now what we're going to talk about is the fact that we do this unintentionally as well. So the classic example that some of you may have heard is industrial melanism in the peppered moth here. So in this case, during the Industrial Revolution, a lot of the trees got covered with black soot, and you see an increase in this dark form of this moth because it more camouflages against the dark trees. And then when the Clean Air Act comes along, you see a decrease in the dark form because it no longer camouflages. So we didn't intend to apply selection pressure there, but we did, and we changed the traits we see in nature. there's some other really cool examples here. So for example, these cliff swallows sometimes nest near highways. And it's been shown that they've evolved shorter wings when they live near highways to veer away from oncoming traffic. These blue tits over in the UK have evolved longer bills to access bird feeders. And these cute little anoles here in Puerto Rico have evolved stickier toe pads to cling to impervious surfaces where you're more likely to find in the city than in nature. So in many ways, we've altered evolutionary trajectories of organisms in nature. I think this is a really tangible example of climate adaptation. So this is work coming out of a group in Montana, and these two cute little guys are both snowshoe hares. They're the same species. Snowshoe hares are brown, but in the winter they molt to white. We can go a couple. Yeah, you can just go up. Yep, that's good, thanks. So in the winter they molt to white, and that's cued by a photoperiod. So it's a seasonal molt. And theoretically, this is adaptive because in the winter there's snow on the ground, and so you're more camouflaged, less likely to be eaten. And what we see when we look at that map is that generally places that are a little warmer or have less snowpack are more likely to have this brown form. So what I didn't tell you is that not all snowshoe hares molt to white. Some stay brown year round. And so that particular trait where you don't molt to white has shown up several times in warmer places where you have less snowpack. And it's been shown that if you mismatch your background, you're less likely to survive. So if you're brown when it's snowy, or if you're white when it's not snowy, you have a 7% decrease in survival. So you can imagine the implications here for climate change. If you have decreased snowpack, but still have the traits where you're molting to white, you're likely to see declines in these populations. And there's been some really great modeling work showing that this is happening in these populations. And this isn't just an issue for the snowshoe hares, although they are really cute, and of course we care about them. There are several different species that have this seasonal molt from white to brown, and are likely to suffer these same mismatch penalties. So I think I like to show that because I think it's a really tangible example of a trait that's visible and associated with climate change. But we don't always have that trait. So I come back here to the example of the mussels. Some of them lived and some of them died, but we don't know why some of them lived and some of them died. So in my lab, what we do is use DNA sequencing to try to figure out some marker for why some live and some die, why some organisms are more tolerant to all the selective pressures from climate change, and why some are more susceptible. So for example here, you might have, so here's our toy example. Again, like bring your brains back to high school biology. We represent DNA by these strings of A's, T's, C's, and G's. And at almost every base pair in the genome, we're all exactly the same. So humans, about 999 out of 1,000 of these locations, we have the same base pair. But that 1 out of 1,000, you have something different, and mussels are the same. So what we do is we look for those differences. Those differences encode the things that are differences among organisms. So all of the different traits that we express. And so we look for those differences in the genome, and then we try to explain responses that we see in nature based on those differences in DNA. So if we look at our mussel example, we might see that some mussels that live, maybe some mussels that live have a C at a particular place in their genome, while maybe the mussels that die have an A. This is a cartoon example, but this is the type of pattern that we would look for. But we're not going to talk about mussels today. What I really want to talk to you about today is some of our work on coral reefs. So corals are a really important ecosystem, both for biodiversity and for humans. So they house a large amount of the biodiversity that you see in the ocean spends part of its time on coral reefs. And that's because they make this really elaborate three-dimensional structure that has shelter for many different organisms. They're also really important for people. So for people in coastal communities, coral reefs provide coastal protection from storms. Because so many fish depend on coral reefs, they provide a source of protein for coastal communities. They also provide dollars from ecotourism. Now, anyone know, is coral a plant or an animal? Ah, thanks, yeah. So one coral is actually millions of tiny animals, millions of genetically identical animals that we call polyps. And you'll see a picture here. And inside those polyps, inside the tissue of the polyps, is a little algae that is photosynthetic. Yeah, it looks like that. And so those photosynthetic algae photosynthesize, meaning they turn the sunlight into nutrients, and they provide those nutrients to the coral. So there's this sort of friendship between the coral and the algae, and it turns out that that's a pretty sensitive friendship. When the environment changes, the coral expels the algae. So you may have seen some headlines about coral bleaching over the last 5 years. It's been especially severe, that when the coral expels the algae, it turns this sort of stark white color. And the problem with that is that the coral's no longer getting the nutrients from the algae. And so functionally, when bleaching happens, the coral has a decent chance of starving to death. And so that's when mortality happens. So bleaching isn't mortality, but bleaching can lead to mortality. But bleaching susceptibility varies a lot in corals. So it varies among species. This is a picture I took in American Samoa about 5 years back. And you see two individuals here. They're right next to each other. And we had just had a recent heat wave event. And you see that this individual here is almost completely bleached, and the individual right next to it is completely healthy. And you can even see some sort of branches covered in algae that used to be part of a coral that is now totally dead. So we see these really different responses to a heat wave event, and we want to know why. And the reason we want to know why is because we want to be able to predict what will happen at the scale of coral reefs in the future. And we start that by looking at the individual corals. So the place that I'm going to take you to is one of my favorite places in the world, Ofu Island in American Samoa. It's actually a US national park. So those of you who collect national parks, I highly suggest it. And the reason that it's really a great place to study this sort of climate change resilience, or resilience to future ocean warming temperatures, is because the structure of the reef, and you can see it a little bit in this overhead image here, creates these pools, and the pools have really different temperature regimes. So you can see on the graph here the temperature in some summer months, and we have the warm pool, which has these huge oscillations. It reaches, 32, 33 degrees Celsius. We've even clocked it at maybe 36 degrees Celsius on a really warm summer day. That's pretty hot. The cool pool has the same oscillations, but they're a lot more dampened. So that pool doesn't reach those high temperatures, but the coral assemblages in both of the pools are pretty healthy. So we're using the warm pool as sort of a proxy for what the ocean might look like in the future. Okay. Yep, so what we want to do is look at the warm pool and the cool pool, and we want to look at the corals that are in the warm pool and the cool pool, and see if we see differences at the level of both the traits, so remember, diversity is the key to adaptation, and at the level of the DNA. So our hypothesis would be that we have some corals in the warm pool that are different in some way than corals that are in the cool pool. That's important for two reasons. The first is that maybe adaptation has already happened in the warm pool. And so that's, again, a proxy for what we might see in the future. And then the other reason that's important is because that's creating the diversity that this population might use to adapt in the future. Okay, so how do we test thermal tolerance in this system? It's pretty laborious. So that's me sampling a coral there. We can cut just little tiny branches off the coral. And then we have these beer coolers that we rigged with like an aquarium system. Yeah, they look like that. So we put aquarium heaters and chillers in there. We've got aquarium lights up. We've got aquarium pumps, but they're really portable because they're beer coolers. And we put those coral pieces in them and we heat them up and we see what happens. And these are some of the different outcomes we might see. We might see that the corals completely bleach. That's that bottom picture where they look white. Or we might see that corals don't bleach at all. So in that way we can explicitly test how tolerant are you to higher temperatures. But I think you've all heard the phrase nature versus nurture. We want to know whether this is their temperature tolerance is due to their recent experience with temperature or whether it's due to their genetics. We want to know whether selection has happened in that warm pool. And to do that, we need to move some stuff around. So what we really want to know is if we take those cool pool corals and stick them in the warm pool, will they learn to be more tolerant? Or is there just something in their DNA that makes them less tolerant? So we do these transplant experiments where we grow out corals, both from the warm pool and the cool pool, in both pools. And they look like this. This is maybe after a year or so of growth. And then the next picture is going to show you after a couple years of growth. So you can actually see quite a bit of growth over a few years. And we can measure that growth as well as thermal tolerance to look at the performance of the corals over time. Okay, so just to show you another way, we have these warm pool corals and cool pool corals, and we're gonna swap them between their pools and see what happens. All right, so this gets a little bit data heavy, so bear with me here. Again, we're starting with corals that came from each of these pools, the warm pool and the cool pool. And then because we're swapping them, but also growing them back in their origin pool, we have all possible combinations here. Yeah, so warm pool corals growing in the warm pool and the cool pool, etc. So first, let's look at tolerance. So that's that thermal tolerance that we measure in the beer coolers. And what we find is that corals from the warm pool always are more tolerant than corals from the cool pool, regardless of where they're growing. So this tells me that there probably has been selection. There's something innately different about corals in the warm pool than corals in the cool pool. They're always more thermally tolerant. If we look at survival of transplant, we see the same thing. Corals from the warm pool are always better at surviving than corals from the cool pool. So they seem sort of all around just tougher. But we see something really different when we look at growth. And that is that corals in the warm pool always grow better. So everyone grows better when it's warm. That makes sense. That's sort of a general rule for living things. Metabolism is faster when it's warmer, so things grow better. Until they don't. Okay, so how is this happening? These pools are really close together. How does natural selection happen in such a small system? Well, this is what we think happens. So up there you have coral babies, our mixed larval pool. Coral babies are called larvae. And corals broadcast spawn. This means that they release their gametes into their water column, hope that their gametes meet up with some other gametes, and make this floating baby that settles somewhere. Yeah, very low parental investment there. All right, so once you have this mixed pool of larvae, of coral babies, they have to settle somewhere. And they settle pretty randomly. So they might settle in the cool pool, they might settle in the warm pool, and you'll probably have a pretty similar assemblage. But then time comes on, maybe you have a heat wave event, and maybe some of those corals in the warm pool that aren't so adapted to living there die off. And so over time, you end up with this different assemblage in the warm pool than in the cool pool, where in the warm pool you have more tolerant corals that survive better, and in the cool pool you have less tolerant corals. And again, we use genomic sequencing. So the problem with this is that all of that experimentation to find out who's a better survivor and who's more tolerant is a lot of work. So when we're thinking of scaling this to predict what will happen to coral reefs in the future, we can't rely on doing tons of experiments on every individual coral. We need a better way to assay tolerance. So we look at DNA, and when we compare corals that are tolerant to corals that aren't tolerant, we find a lot of differences, actually. We find differences in DNA sequence at over 100 different genes. So many different genes are associated with thermal tolerance in coral. It would be easier if there were one, but there are lots, so lots of differences. So now we're going to use these DNA sequences and see if we can do some prediction. And for that, we need to go to a totally different system because we know the OFU system already. Any prediction that we do in the OFU system would not be independent. So we already did our experiments in the OFU system, and what we found was some genes associated with thermal tolerance. We're going to go and use that information to try to run some predictions. And to do that, we're going to take it to a new system, this island called Rarotonga. Rarotonga is in the Cook Islands. It's quite a bit south of American Samoa. And in fact, it's sort of toward the southern edge of coral growth in the open ocean, which means for all intensive purposes, this is like a cool water tropical reef, as cool as tropical reefs get. And this is important when we're thinking about adaptation. We're wondering, first of all, do the corals in these cool water reefs even have these thermal tolerance genes? Does that variation that we need for natural selection exist? And how might that change over time? So we're going to use some computer simulations to start with our DNA sequences and simulate future population sizes. So what is the future of these coral reefs? First, we need to know what the temperature future looks like. So luckily, we have some really good predictions of what the future temperature looks like. And these are based on emissions scenarios from the IPCC. These are actually the old emission scenarios. There are new ones now, but this will do for us. So the ones you see in blue are CP 2.6 and 4.5 are more mitigated emission scenarios. And when you look at what that does to the temperature in Rarotonga, you don't see a lot of increase. So the sort of hottest temperature is about 28.5. It doesn't change a lot in those mitigated emission scenarios. But when you look at the more severe emission scenarios, 6.0 and 8.5, 8.5 is what we used to call business as usual. That's the trajectory we're on now. You see about a 2.5 degree increase by the end of the century. So the waters in Rarotonga are 2.5 degrees hotter by the end of the century than there are now. Most corals right now live within 1 to 2 degrees of their upper thermal limits. So for corals, this is not good. Okay, so let's see what our simulations do. Again, this is based on what we know about the genetics of corals. Under the mitigated emission scenarios, so I'm showing you population size here, and then over time, you see that we have persistence of the coral reef. So you see some decline in how many corals that we have, but we still have corals. On the other hand, under those more severe emission scenarios, we see extinction of those populations, and sometimes quite quickly. So in that business as usual scenario, you see extinction of the population by the middle of the century. And a very sort of sad anecdotal note to this is that this initial sampling was done in 2014. In December, I visited these same reefs, and coral cover is much lower than it was in 2014. So it does seem like we're on some of those downward trajectories. Okay, so we know that coral reefs globally are suffering. And people are really concerned about this and have been trying to think of mitigation strategies to preserve coral reefs. And one of the popular ones right now are these coral nurseries, where you could potentially grow out corals and plant them back out on the reef. And you could even grow out corals that you think are thermally tolerant, either because you've tested their thermal tolerance or because you know something about their DNA sequence, and preferentially plant those out on the reef. And the idea is that that would speed up adaptation. So if you think back to like the mouse example, it would be akin to adding more black mice to that population so that the frequency of that adaptive trait is higher. So that when those black mice reproduce, the population is better adapted to their environment. So what we did was to do some more simulations, and this time we simulated adding corals that had DNA indicative of thermal tolerance. So this is the same curve that you saw on the last slide. We're using RCP 6.0. This is not quite business as usual, but one that still went extinct. And what we're gonna do is add either 25 or 50 corals per year. And these are, again, thermally tolerant corals every single year to our population and see what happens. So what we see is that if we add those corals every year, the reef persists to the end of the century. You see fewer corals by the end of the century, but the reef is still there, which is a very good thing. However, so at the surface of this seems really good, right? We want to keep corals around. Here's a mitigation strategy. Our computer models say that it might work. I do want to put this into a little bit of perspective. Adding 10, 25, or 50 corals to the single reef is like adding 1% of the population size every year until the end of the century. That's a huge amount of human labor, not to mention the huge question of where we get those thermally tolerant corals from. So there is a mitigation strategy. It does seem to speed adaptation, but there's some huge scaling and logistical questions in terms of actually making this into a conservation strategy. And then I just want to kind of end with thinking of this concept of survival of the fittest. I think it's easy as, maybe not easy, but I think as a fairly optimistic person that works in the field of climate change, it's easy to look for wins. And when we think of natural selection, the obvious thing to think is, well, adaptation will help. Adaptation will change populations so that they're better adapted to future climate change, and then everything will be okay. However, there is a downside to this, even if populations do adapt, and even if we have those populations in the future. And that's because in nature, trade-offs are common. Trade-offs, in fact, are the rule. Biodiversity exists because there are trade-offs. Diversity exists because there are trade-offs. And we see that in our system too. So in this particular case, we see that corals that are better at surviving have lower growth rates. So they're slower growers. So those really tough corals that are tolerant to heat waves and that survive everything are really slow growers. And for corals, that can lead to lower reproductive output. And so this isn't necessarily a good long-term solution. Although those corals might survive to the end of the century, they might not build reefs as quickly. So when we think about adaptation and climate change, I think it is a good lesson to think about nature and its resilience and adaptation as part of that resilience. But I also want to think about what we're losing in the process of adaptation. So what did those bleached corals have that the unbleached corals didn't? What did the dead mussels, what traits did those dead mussels carry that the live mussels didn't? Or these cute little white snowshoe hares? What other traits did they have that might have benefited them in the future? For example, some unknown selective pressure rips through these populations, maybe a disease, maybe the climate shifts again, and they don't necessarily have the diversity, the variation to adapt again. So just going back to this idea, diversity is the key to adaptation. Adaptation can help, adaptation can change the traits that we see, and they might be better suited to their environment, but that does have limits. And it's better to think of conserving diversity overall than any necessary one trait. And with that, I'd like to thank all of you for coming today. This is such a fun event, and I'm so honored to be able to speak to you tonight. All the folks up there were people that worked with me on these studies, as well as funding agencies that funded them. You can see some of my current lab members here doing continuing work on coral. My lab studies coral, but a bunch of other things too. If you're interested in learning more, you can either go to our website, or I made this fancy QR code. You can use that too. Or I'd be happy to talk with you about any of the other work that we're doing in the lab. Otherwise, thank you. I'm happy to chat more about if you have any questions.
I'm sure everybody has some questions. Does anybody have any questions? Can we get some house lights up, please, for
Sure.
And Doctor, we have time for questions. Oh, there we go. Oh, it's a good-looking group. I had no idea. Much better.
Do you ever see any kind of species in which the male or the female adapts faster, better, slower? Difference,
Yeah, that's a great question.
including humans, by the way.
I don't know that much about humans, and I'm, a little bit wary of wandering into that area. But,
Please do. I prefer women, so to speak.
but, so for corals, no, because most corals are hermaphrodites. They're both male and female at the same time. But in some other species, and now I'm not totally remembering which, you do see thermal tolerance differences between males and females, for example. An interesting and kind of related point is like animals that do temperature-dependent sex determination, so like turtles. This is a huge problem with climate change. If sex determination is dependent on temperature, you end up getting really skewed sex ratios. So for example, I think it's the case where some entire turtle populations become female because it's too warm, and all of the turtles that are hatching are female because their sex depends on the temperature when they hatch. Yeah, so it is a problem for those sex ratios for sure. Yeah, Nina?
Are there experiments similar to what you're doing with food kinds of things for humans?
Food?
Yeah.
Can you repeat the question?
Oh yeah, so the question was, are there similar types of experiments for food? And the answer is yes. There's lots of people that are very concerned about the sustainability of our crops under future climate change and doing experiments about not only whether our crops will be able to live in the places they live, but also particular varieties that might be more resilient to climate change.
They're looking at the genetics.
Yeah, lots of crop genetics. In fact, that's one of the larger funded sort of genetic fields.
So what about bananas? I heard there's a problem.
I don't know that much about bananas, except that there's no genetic variation. They're all totally sterile. I don't actually like bananas, so I'm okay with it.
I told you, don't bring up bananas.
Is this like a pattern? Yeah.
So, the great barrier reef off Australia is noted for its bleaching on that? What's the potential success rate of recovery? And I know, having died there also, that like they just say that the Asian, they just had like no consciousness and they're walking over everything and they're trying to cut back how many visitors. Your thoughts on that?
Yeah, so there's two issues there. So the question was the Great Barrier Reef has shown a lot of decline over several years, and that's not only due to climate change, that's due to the impact of heavy tourism. And that's true. There's been a lot of tourist impact on the reef. I would say the impact from rising temperatures is a lot more. For example, there was a global bleaching event 2015 to 2018, and half of the corals on the Great Barrier Reef died in that short period of time. So that's way more damage than any tourist is doing. However, stressors combine, right? So there is a little bit to think about in terms of ecosystems that are already a little bit fragile and how we treat those. There is some evidence showing that corals that are in more pristine reefs are less likely to bleach during these warm events. So it's possible that, for example, in some of these more remote locations, you might see coral reefs that are more likely to persist. But that research is still pretty preliminary. Yeah.
Coral's been around for a long time, I'm assuming billions of years.
Mm-hmm.
The question is obvious, how has it survived? The temperature has gone up and down. Is it because of the drastic change in nature of it, or has this drastic change occurred in the past, and is there some mechanism that still might be out there that we don't know about?
Yeah, that's a fantastic question. So the question is, corals have been around for millions of years, correct? And what is different about now, and will they live through it? So I have no doubt that corals will live through climate change. The question is, will coral reefs live through climate change? And while corals have been around for millions of years, these sort of photosynthetic coral reefs, these communities we see right now, have not. And those are the ones that are most sensitive to bleaching. And so that symbiosis with the algae is both good and bad. It turns out that what happens is that when bleaching happens, the algae starts to turn some of those photosynthetic products toxic, and that's when the coral kicks them out. So corals, in some form, will live through climate change, but whether they create these coral reefs that are habitats for fish and invertebrates, maybe not. Yeah?
I've heard, I think this is correct, that plankton is critical in the ocean food chain. So what do we know about tolerance and adaptation of plankton?
Yeah, I don't actually know anything about tolerance and adaptation of plankton. I've seen papers come across like my Google searches about them, and so I know that people are working on looking at changes in plankton abundance that are associated with warming, but if I told you anything, I'd be making it up. Yeah.
Yes, do you see much chance of corals migrating or being introduced to slightly cooler areas of the ocean?
Yeah, so the question was, is there any evidence that corals are either migrating or being introduced into cool areas? Yes. There's one situation that I know of where that's happening. So we call that a range shift, where because the overall global temperatures are rising, organisms might move away from the equator into places that sort of are the same temperature as the place they used to live. So in Japan, we see northern movement of the coral reefs. And in Japan, they actually consider this an issue because they have macroalgae, so like our kelp forests, that are the dominant ecosystem there, and they're really important ecosystems as well. So in that particular location, the northern movement of corals is a bit of a problem because they're outcompeting the local kelp forest system. I don't know of any other places. The problem with corals is that their habitat is super patchy across the ocean. They can only live in these pretty shallow areas, which means mostly around islands or atolls. So it's hard to document much movement, but in some places they do. Yeah.
In in the vernacular, talk about Lamarckian evolution in human populations because of PSCT, and those are passed theoretically to children. Is there any experimental evidence that shows that's happening in corals?
Yeah, so the question was, Lamarckian evolution, is it happening? In corals specifically. And so, as an example, this gentleman gave, like human stress, we know, is passed down through generations. And so the mechanism for that is something we call epigenetics. And that is tags that get put on DNA that are not changes to the DNA sequence itself, but they're little tags that get put on the DNA, and that can be inherited between parents and offspring. It's not as much of a permanent solution as differences in DNA sequences. I actually have a graduate student that's working on that question, looking at these epigenetic tags in corals, and looking at whether or not they're associated with thermal tolerance. There is some evidence that that different epigenetic profiles are associated with not thermal tolerance, but how much you can change your thermal tolerance. So we talked about that nature versus nurture. So some corals are able to be a little bit more flexible with their thermal tolerance than others, and it seems like epigenetics probably plays a role there. There's just fewer studies on whether epigenetics are associated with thermal tolerance overall, and we actually don't even know if in corals those markers are inherited like they are in animals, or in mammals. Yeah?
How much effect has fishermen's nets have on survival of corals?
The question was, how much effect does fishermen's nets have on survival of corals? So fishing pressure in general does have both direct and indirect effects on coral reefs. So what I mean by that is that, yes, direct damage to coral reefs is bad. There's also indirect effects because corals depend on the entire community, and it turns out that pulling out key contributors to that, like some of the dominant fish, is bad for the corals as well. I don't know if net fishing is such a big deal. There used to be more practice of things like dynamite fishing that were quite destructive to reefs. But I think most communities are pulling back on that with, as we become more aware of the threats to reefs. Yeah?
You mentioned that they're starting coral nurseries, and I was wondering if instead of that, you could just take the gametes and spread them across the coral.
Yeah, so the question was, I mentioned coral nurseries. Is there an alternative to that where you could just sort of collect the gametes and spread them around? Yes and no. So the problem with that is that it's really hard to get the corals to spawn in captivity. So people are doing it, but it's really challenging. So this is why most coral nurseries, rather than making their own gametes, they just break up the corals and grow the pieces. However, there are some coral nurseries that are trying to increase wild spawning. So it's not exactly what you're talking about, but what they're trying to do is make those fragments and increase the capacity for those fragments to spawn themselves. Most coral babies die. That's just a characteristic of the coral life cycle that's really normal. And so even if we put thousands of coral larvae out there, most of them would die. So the advantage to taking the adult and fragmenting it is it has a much greater chance of survival. This is awesome.
We got two more.
All right.
What is the status of the mussels on the North Coast now?
Yeah, that's a great question. I don't know the answer to that question, and I should have asked because a collaborator of mine provided these photos.
What's the question?
Oh, sorry. What is the status of the mussels on the North Coast now? In general, mussel beds are pretty productive, so I wouldn't be surprised if they come back pretty quickly, but I don't know for sure. One more question? Yeah?
When you compared the genomes of the warm pool and the cool pool, and you found some 100 genetic differences, how were you able to determine that which of the 100 were, due to thermal selection?
Yeah, that's a great question. So the question is, when we compare the genomes in the warm pool and the cool pool, and we want to find genes associated with thermal tolerance, how do we know they're associated with thermal tolerance? This is correlation, this is not causation, so there is a little bit of a caveat there, but we do a few things. The first is that in addition to just comparing between the pools, so we know that the warm pool has higher thermal tolerance than the cool pool. So the sort of broadest scale thing we can do is just say, where are the genetic differences between the two pools? But we can also measure temperature for every single individual coral in those pools. And so we look at the corals that are living in the hottest places in those pools and the coolest places in those pools, and we compare those. And the third thing that we can do is compare them with our experimental thermal tolerance tests. So when we put the corals in the beer coolers, we can actually measure who's most thermally tolerant and associate that with the DNA differences. Does that make sense? All right, where are we at?
One more?
One more?
So the nursery idea seems to be using the heavy hand of artificial selection by putting the genes you want into the population.
Correct.
But following your theme of the importance of diversity, is there another strategy that just pumps up the genetic diversity in the population, hoping that it will stumble upon good solutions that way?
Yeah, so different nurseries are managed in really different ways. Some of them have the goal of strictly increasing coral cover. So in that case, we don't care what the DNA of these corals look like. We're just trying to plant as many as we can. And so that might increase diversity overall. Some nurseries are actively artificially selecting for thermal tolerance, even to the point where they might be breeding. Some people are even talking about genetically modifying corals in order to be more thermally tolerant. You might guess where I stand on that based on the last part of my talk. But I think there's also more attention being paid to how we might intentionally increase diversity. So it's sort of a more portfolio-like effect in order to select not only thermally tolerant corals, but a diversity of corals. However, despite like my worry about not conserving diversity, I will say that like some corals are still better than no corals. So right now is a pretty severe situation for corals. And if we have some, even if our diversity is decreased, it's still better than no corals at all.
Thank you.
All right, thanks so much, guys.
Dr. Rachael Bay, everybody. Thank you very much. Rock star right here in our town. How about that, huh? Isn't that great? Great questions. Thank you. You're a terrific crowd. I'm just so proud to be associated with the community. I think you're a little impressed with our questions in our group. Isn't it terrific? It's great, and it's thrilling. Thank you very much to everyone joining us today for the 26th annual Sacramento Darwin Day celebration. As I mentioned in my opening remarks, the Sacramento Darwin Day Organizing Committee is composed of a group of dedicated and passionate volunteers focused on promoting and supporting science in the greater Sacramento area. Each year, the group seeks out an exciting and compelling speaker, and I'm so proud to have helped bring Dr. Bay to you today. I especially to thank Minga and Bronda, who did the searching and found Dr. Bay for us. And again, thank you, Dr. Bay, for agreeing and bringing your wonderful husband tonight. It's really been a treat. Before we close, I'd like to encourage each of you to think about how you can support this fun and important event. Perhaps you can help us meet some of our operational goals and objectives by volunteering your services. Special thanks to the kitchen crew, Renee and Marcus. You guys have been fantastic. Marcus is probably already down there getting our party ready, who make the event so fun each year. Each of you have shown your appreciation with your ticket purchase, and many have donated more, and I want to thank you. Many of you have invited people and turned them on to what we are doing. This is the best way for our event to grow, and I encourage each of you to find a friend and invite them next year. It is my stated goal as chair to not only try and produce a top-notch annual event, but to lead the event forward into the future. This means an increased focus on diversity and inclusion. It's our stated goal to make all science enthusiasts aware of our event and to make them all feel welcome. If you have any ideas how we can continue to pursue this goal, we are all ears. Let me take a moment to reflect on how far Sacramento's science community has come. Mosac has been transformed from a once neglected electrical powerhouse into a fully functioning powerhouse of science education. See what I did there, Minga? Thanks to the inspiring leadership of Michael, Michelle, Jennifer, Rod, and Susie, who's downstairs getting ready as well, and of course Ford. I butchered your name earlier. Thank you again, Ford. Thank you. I ask you to consider donating and volunteering so the youth in our community will know that adults in their world understand and appreciate the pursuit of knowledge. I'm a volunteer here. It's very easy and it's a lot of fun. It has been a challenging couple of years, and I am sure new challenges will present themselves. The way forward for the world can be found in the roots of the community here today. By working together, supporting and following the science, we can ensure the very best chance possible for the survival of our species and the world. Let's let science lead the way. Let's let truth lead the way. Let us lead the way. Thank you. It has been an amazing and rewarding evening, and I cannot thank you all enough for your support and friendship. Thank you again to Dr. Bay, Minga, and the committee members. Please enjoy the museum, and be sure to stop by the donation table downstairs and the museum shop to pick up your gift, a small memento of a giant of science. And with that one, and we're going to sing happy birthday. Until next year we meet again, please take care of yourselves and each other. Thank you again. Have a great night.